There are many blood types in the world. Many people think there are only 8 blood groups, whereas there are actually more than 30 different and rare ones. We will explain them all in a simplified way in this video. We will begin with the well-known major groups. O positive, 38%, A positive, 27%, B positive, 20%, A B positive, 5%, O negative, 7%, A negative, 1.5%, B negative, 1.5%, A B negative, 0.6%. Now the special blood types. Kell blood type, the third most important system. Most people lack the Kell antigen, K, making the transfusion of compatible blood a process requiring extreme precision. Duffy blood type, a variant that is rare in the West but very common in Africa, characterized by providing natural genetic protection against malaria. Mumbai blood type. In analysis, it appears to be type O but lacks the fundamental substance H antigen. It affects 1 in every million people globally and 1 in every 10,000 in India. Rare Lutheran blood type. A very rare mutation in which the individual completely lacks Lutheran system antigens, making it extremely difficult to find compatible blood. Sarah blood type. A unique and genetically isolated lineage. It has been officially recorded in only 2 families worldwide. The golden blood type. It is the rarest and most expensive on the planet. The individual possessing it lacks all 61 antigens of the Rh system. Only about 50 such cases have been recorded in all of modern history. GE null lack 3 major antigens found on the red blood cells of other humans. If a person with this blood type receives a transfusion of ordinary blood, their body mounts a violent immune attack. Very rare cases of this blood type have been recorded worldwide, and it is closely monitored by international blood banks. The Diego blood type. This blood group is quite unusual, as it is sometimes used in anthropology, the study of human populations. The DHA antigen is extremely rare among Caucasian and African populations, less than 1%, yet its prevalence rises to approximately 36% among certain indigenous tribes in South America, Native Americans, and some Asian populations. A crisis arises in cases of interracial marriage, as the pregnant mother's body may attack the fetus if it inherits this rare blood type from its father. The Colton negative. Colton antigens are located on a crucial protein found in blood cells, known as aquaporin 1, which is responsible for balancing water and maintaining the shape of red blood cells. Some individuals are born with a mutation that results in a complete absence of Colton antigens, CoABS, a condition so rare that finding a matching donor requires international coordination between frozen blood banks. Philly, it is classified as one of the most difficult rare blood types in blood banks. It is very difficult to find a VEL-negative individual. The prevalence is approximately 1 in 4,000 people in Europe, and even lower in the rest of the world. Individuals lacking the VEL antigen produce a highly potent antibody, anti-VEL, upon exposure to ordinary blood, causing immediate and direct destruction of the transfused red blood cells. Langris and Jr. The genes and proteins responsible for these 2 systems were precisely identified only in 2012. The rare junior blood type is particularly prevalent in Japan compared to the rest of the world due to genetic and societal factors, and individuals with this type face extreme difficulty if they require a blood transfusion outside their country. The rare Okites. It belongs to a system known as OK. The vast majority of humans, more than 99.9%, carry the OCO-high antigen on their red blood cells. Individuals with this extremely rare blood type lack this specific antigen. First discovered in a Japanese female patient, this blood type poses a significant challenge. If a pregnant woman conceives a child who has inherited the antigen from the father, as the mother's body begins to attack the fetus's blood, the Ogier blood type. This system was registered globally following the discovery of a case involving a woman of African descent whose blood was found to lack an antigen, known as AT-CARE-A, that is very common among humans. Globally, the prevalence is virtually non-existent outside of very specific families of African descent. Individuals with this type produce potent antibodies that reject any conventional blood. The Cromer. Negative blood type Cromer system antigens are associated with proteins that protect red blood cells from being mistakenly attacked by the individual's own immune system. Individuals born with a mutation resulting in the complete absence of Cromer antigens, CRA-, are extremely rare and require highly specialized medical care regarding blood screening and cross-matching prior to surgery. The rare MNS subsystem. It is an old blood group system, discovered in 1927, that comprises more than 50 antigens. This subtype represents an exceptional genetic mutation in which the individual completely lacks the system's key antigens, such as MNS and S. Their blood cells appear entirely devoid of these common proteins, making their blood a rare genetic condition studied in laboratories worldwide. The rare NOPS system antigens bind to specific receptors on blood cells. The absence of the KNA antigen is extremely rare in white, Caucasian populations, though it appears at slightly higher frequencies in certain ethnic groups in Africa and North America. The rare Shibano. This family belongs to a unique genetic system linked to immune system proteins, specifically a component called complement C4. Some individuals completely lack Cheeto or Rogers antigens, a very rare genetic condition. The rare Indian, as the name implies, the INA antigen, is genetically associated with the population of the Indian subcontinent. This antigen is completely absent in over 99% of the global population, Europeans, Africans, and Americans, yet it appears in approximately 4% of the populations of India and Iran. John Hill. This family is associated with an antigen known as JMH, located on a protein specific to lymphocytes and blood cells. Some individuals are born completely lacking this antigen due to genetic factors, JMHA. Biddulph blood type. It was discovered and named after the Biddulph family. Members of very specific families worldwide possess a rare antigen on their red blood cells that is not found in the rest of the human population. These rare blood group variants cause hemolytic disease in the fetus during pregnancy, as the mother's body, if she lacks the specific antigen, attacks the fetal blood inherited from the father. Low Ernest rare sub. We previously mentioned the complete absence of the Lutheran system. However, there is a very rare variant within this system known as LU-AB+, as well as other minute genetic configurations classified as orphan antigens. Antigens found in only a single family or in a limited number of samples, preserved at the International Blood Group Reference Laboratory in the UK. The rare Forsman. For many years, it was believed that the Forsman antigen was present only in certain animals, such as sheep, and absent in humans. In extremely rare cases, scientists have discovered a very small number of human families worldwide who carry this antigen in their blood, a hereditary trait that renders their blood system unique and distinct from the standard human system. The negative Dombrock. The Dombrock system includes well-known antigens such as DOA and DOB. The complete absence of these antigens, DO_null, is an extremely rare condition. The danger lies in the fact that the antibodies the patient produces against ordinary blood cause the slow, insidious destruction of transfused red blood cells, a process that is difficult for physicians to detect immediately during the transfusion. The rare Gerbish. This blood group lacks Gerbish system antigens. It is extremely rare globally, yet it is found in up to 50% of certain indigenous tribes in Papua New Guinea. Just like the Duffy blood group in relation to malaria, the absence of the Gerbish antigen protects blood cells from invasion by a specific type of malaria parasite, providing the individual with natural immunity. Belzerod. This system was named after a small German town where the first family carrying a unique genetic mutation in red blood cell proteins was identified. This blood group is rarely mentioned outside the context of this very limited genetic lineage. The rare XG. This system is very unique because the gene responsible for it is located on the X sex chromosome. Due to this sex-linked association, the XGA antigen is present in approximately 89% of women, but only 66% of men. Its complete absence is classified as a rare blood type, studied to understand the inheritance of sex-linked diseases. The rare Landsteiner-Wiener. It was named after the renowned scientists Landsteiner and Wiener, discoverers of blood groups and the Rh factor. This blood type is extremely rare, LWA or LW null, and unusual because its antibodies behave exactly like Rh-positive antibodies. a fact that used to deeply puzzle doctors in the past when attempting to match blood.